We all have a freezer. Pretty basic, right? Keep food from going bad. Just put it on pause with cold.
Right. Simple concept.
Now take that same idea, stopping time with cold, but scale it way, way up. Imagine doing that to something incredibly complex and alive, like a human organ, a kidney, maybe a heart.
A whole liver, yeah.
And then not just freezing it, but days later, maybe even longer, bringing it back, fully functional, putting it into someone and having it just work.
For years, that's been pure science fiction territory, hasn't it?
Totally. But the thing is, we might actually be getting close. Really close. Welcome back to the Deep Dive. Today, we are jumping headfirst into the really cutting-edge world of cryopreservation. That's the science, basically, of preserving biological stuff. Could be tiny cells, could eventually be whole organs at incredibly low temperatures.
We're talking about hitting the biological pause button.
Exactly. Stopping biological time, at least at the cellular level.
And our mission today, well, it's to walk you through some really interesting sources we've dug into. We've got a big scientific review paper that gives a fantastic overview of where things stand right now with preserving tissues and organs. Think of it as the technical deep dive.
The state of the art.
Right. And then we also have this news article that just came out announcing what looks like a pretty major breakthrough, a company that seems to be, cracking some of those really tough, long-standing problems.
Yeah, it's a great combination.
Our goal is to pull out the key stuff, connect the dots between the fundamental challenges the science paper lays out and these potential solutions that are popping up. See where we are and maybe where we're headed.
Exactly. So we get the detailed science picture from the review, the difficulties, why it works for some tissues but not others, a look at some cool experimental tech.
The nuts and bolts.
Right. And then this news piece lands showing a real-world demo that just makes you go, whoa. It's like seeing the blueprint and then someone says, hey, we actually built one and it looks like it works.
And putting those pieces together, that's what's really fascinating. Why should you care about this? Well, think about it. Hundreds of thousands of people around the world are on organ transplant waiting lists.
Huge numbers.
Yeah. And think about fertility preservation. Clinics helping people build families when maybe they couldn't before because of medical treatments. Cryopreservation is key there.
Right, storing eggs, sperm, tissue.
Exactly. Or efforts to save endangered species by banking their genetic material. Even things like emergency medicine, cooling patients down after trauma to buy time, protect organs. Cryopreservation, or at least the principles behind it, touches so many areas. It's not just lab work. It holds this huge transformative potential. Understanding it is understanding a big piece of the future.
Okay, let's really break it down. Cryopreservation, what are we actually talking about here? At its core, it's using really, really low temperatures.
We're talking way below freezing, often down to -80 Celsius, or even plunging into liquid nitrogen, which is -196 C.
-196, that's incredibly cold. And the aim is to keep living cells, tissues, maybe organs, structurally intact and crucially functional.
That's the goal. The big, ambitious goal is basically to stop all biological activity, shut down metabolism, put biological time on hold, maybe indefinitely.
Hit pause.
Hit pause. And the purpose, well, it's fundamental to so much modern science and medicine. Biobanking, for starters. You need long-term, stable storage for huge collections of biological samples.
Cell lines for research, patient samples.
Right, samples for diagnostics, maybe tracking how a disease develops over decades. Tissue samples for future therapies like in regenerative medicine. Biobanking underpins all of that. It makes sure we have healthy cells available when needed, maybe years after they were collected.
And fertility preservation, you mentioned.
Absolutely crucial there. Storing sperm, eggs, embryos, and now increasingly ovarian and testicular tissue too. It gives people options if their fertility is threatened by, say, cancer treatments.
Life-changing stuff.
Totally. And then there's conservation, preserving genetic material from endangered plants and animals, a kind of biological arc. And even the principles, like we said, feed into therapeutic hypothermia in ERs. Cooling someone after a cardiac arrest or a bad injury can slow down damage, protect the brain. Now, that's usually warmer temps than true cryopreservation, but the underlying understanding of how cold affects biology is key. Optimizing protocols everywhere depends on really getting this science right.
Okay, so it's a vital tool, huge applications. But let's go back to my kitchen freezer analogy. Why can't we just take, say, a donated kidney, pop it in a box, and stick it in a super cold scientific freezer at -80? What stops that from working?
Yes. This is where we hit the fundamental, really brutal challenge. The biggest, most destructive problem is ice. Ice crystal formation.
Ice. Just like in the freezer?
Well, yes and no. Living things are mostly water, right? And when water freezes, it doesn't just turn solid, it expands. And it forms these crystalline structures.
Sharp edges.
Very sharp edges. Inside the incredibly delicate structures of cells and tissues, these ice crystals are just devastating. Imagine tiny shards of glass forming everywhere, inside cells, between cells, in the structural matrix holding an organ together.
Ouch.
They physically puncture cell membranes. They rip apart the intricate networks of proteins and lipids. They shred tissue architecture. It's catastrophic mechanical damage. And once it happens, it's irreversible when you thaw it out.
Okay, so physical damage from the ice shards. That sounds bad enough.
Right.
But you mentioned it's not the only problem.
Not by a long shot. The damage is really multifaceted. Scientists lump it all together and call it cryo-injury. So, beyond the physical puncturing by ice, you get major chemical and osmotic stress.
Osmotic stress.
Yeah. As water starts to freeze outside the cells, the liquid water that's left behind gets more and more concentrated with salts, proteins, other solutes.
Right, because the water's turning into ice, leaving the other stuff behind.
Exactly. So the solution outside the cells becomes super concentrated, or hypertonic. And what does water do when there's a concentration difference across a membrane?
It moves. It gets drawn out of the cells towards the higher concentration.
Precisely. So the cells start to dehydrate as water rushes out. This shrinking and dehydration itself damages cell components. Plus, the now super high concentration of salts inside the shrunken cell can be directly toxic.
So punctured by ice outside, dehydrated, and poisoned by salt inside? Nasty.
It is, and it doesn't stop there. The damage isn't just during freezing, the thawing or rewarming phase.
Yeah.
That's just as critical, and it can cause a whole new set of problems.
Oh, great. More problems.
Yeah. If you don't warm a frozen sample fast enough, or if the temperature fluctuates, any tiny ice crystals that formed can start to grow. It's called recrystallization. They get bigger, sharper, more damaging.
So even small ice crystals become big problems later.
Right. And if you manage to vitrify something, turn it into glass, without ice, rewarming is even trickier.
Yeah.
If you don't warm it up ultra fast and perfectly evenly, the glassy state can collapse. And then you get massive, sudden ice formation throughout the sample. That's called devitrification. Catastrophic.
It's vitrification. Sounds bad.
It's fatal for the tissue. So scientists are fighting damage during cooling, potential damage during storage if temps aren't stable, and really significant damage during rewarming. It's a minefield.
Okay. So the challenge is somehow get things really cold without letting ice wreck everything and manage all those other chemical and osmotic stresses both going down and coming back up. The review paper we looked at digs into the two main traditional ways scientists have tried to do this: slow freezing and vitrification.
Right, those are the kind of classic approaches. Two different philosophies, really, each with its own pros and cons. Slow freezing is maybe the more intuitive one.
How does that work?
You cool the sample down very, very gradually. Typically around 1 degree Celsius per minute, maybe even slower. You take it down to really low sub-zero temperatures, and then usually store it in liquid nitrogen at minus 196 C.
1 degree a minute, that is slow. Why so slow?
The idea is to control where the ice forms. As the solution outside the cells starts to freeze slowly, the solute concentration goes up out there, just like we discussed.
Right, drawing water out of the cells.
Exactly. The slow cooling rate gives the water time to move out of the cells via osmosis before the inside of the cell gets cold enough to freeze. So you get controlled dehydration inside the cells, which makes them less likely to form damaging ice internally when the temperature drops further. Controlled slow freezing, usually using special programmable freezers, is pretty standard for many types of cells and simpler tissues.
Okay, that makes sense. And looking at that comparison table in the paper, table 2, it seems like slow freezing has some points in its favor, especially for smaller things.
It definitely does, for certain jobs. The table mentions it's generally easier to handle technically compared to vitrification. It's also very controllable and reproducible because you've got a machine precisely managing that cooling rate.
Consistency is good.
Very important. And crucially, it usually needs lower concentrations of those cryoprotective agents, the CPAs, the antifreeze chemicals you add.
Ah, the CPAs. We'll come back to those. Lower concentration is better, I assume.
Much better,
Yeah.
because CPAs are toxic. We'll definitely get into that. Lower concentrations mean less toxicity. Plus, with lower amounts, they can spread more evenly through the sample and get into the spaces around the cells properly before things start freezing. For single cells, or small groups of cells, slow freezing often gives really good survival rates after thawing.
But there's always a but, isn't there? The downsides look pretty major if you want to freeze anything bigger or more complicated than just cells in a dish.
That's exactly where it hits its limits. The main drawback is, while it tries to avoid ice inside cells, it still relies on ice forming outside the cells. And that extracellular ice still causes physical damage. It still drives that osmotic stress and dehydration. It's still a big part of cryo-injury.
So you're still getting ice damage, just maybe not inside the cells as much.
Pretty much.
Hmm.
Table 2 also notes it can be expensive because you need those specialized controlled rate freezer machines. And the whole process takes a long time, often more than 3 hours for one batch. That's a bottleneck if you're trying to process lots of samples.
3 hours per sample?
Yeah.
Wow.
And maybe the biggest limitation for what we're talking about today, slow freezing generally only works well for very small samples if you're dealing with complex tissues or organs. Even when it's optimized, you can still lose a lot of cells. Studies mention up to 30% cell death is possible.
Okay, so slow freezing manages ice by pumping water out, but still has ice issues and doesn't scale well. What about the other one, vitrification? That sounds glassy.
It's a totally different strategy. Vitrification aims to prevent ice formation completely. Forget managing it, just avoid it altogether.
Oh.
By using very, very high concentrations of those CPAs, we're talking maybe 40, 50, even 60% or more of chemicals like glycerol or DMSO, and then cooling the sample extremely fast, often just plunging it straight into liquid nitrogen.
Whoa. High antifreeze, super fast cooling. What's the goal there?
The goal is to bypass ice formation entirely. You turn the water CPA mixture into a solid, but an amorphous non-crystalline solid, a glass. Think of it like cooling it so quickly and with so much antifreeze mixed in that the water molecules just don't have time to arrange themselves into that ordered crystalline ice structure. They get locked in place in a disordered liquid-like arrangement, but solid.
A solid liquid.
Kind of. A glassy state. The high CPA concentration replaces a lot of the water and drastically lowers the freezing point, making it possible for the rapid cooling to essentially outrun ice crystal formation and reach that glassy state.
Okay, so the big advantage. Potentially no ice damage at all. That sounds great. But what's the catch? Those high CPA levels and super fast cooling must cause new problems.
Oh, absolutely. That's where the major trade-offs kick in. Vitrification's main theoretical plus, as Table 2 points out, is that low risk of ice damage, if you do it perfectly. And it's fast. The cooling part can take less than 10 minutes, plus maybe cheaper equipment-wise, since you might not need a fancy machine, just the liquid nitrogen. But the massive challenge is the toxicity, those super high CPA concentrations needed. They're really harsh on cells. They can mess up cell functions, damage proteins, disrupt membranes, especially if the cells are exposed for too long.
Right, the toxicity problem again, but amplified.
Exactly. And handling these thick, syrupy, high CPA solutions is tricky. Getting them evenly distributed throughout a tissue, and then achieving that ultra-rapid cooling uniformly across the whole sample. That's really hard, especially for anything bigger than tiny.
If it's not uniform.
Big problems. If parts don't cool fast enough, you get ice inside the cells, exactly what you tried to avoid. If parts don't warm up fast enough and evenly during rewarming, you get that catastrophic devitrification, massive ice formation. Plus, you still have osmotic stress issues when you're adding and removing those highly concentrated CPAs. And the rapid cooling itself can cause thermal stress, making the brittle, glassy sample literally crack or fracture.
Fracturing. Yikes. So it's delicate and only works for tiny samples usually.
Typically, yeah. The volume you can successfully vitrify is usually minuscule, like a few microliters. People have developed specialized systems, open or closed containers, to handle slightly larger volumes, but open systems risk contamination. It's really challenging to scale up.
So it feels like we're caught, aren't we? Between the ice damage and slow freezing and the toxicity and physical stresses of vitrification, especially when we start thinking about something the size of an organ.
That's the dilemma.
And the way we actually preserve organs for transplant right now, the standard method doesn't even try to do either of these, does it?
No, not even close. The current clinical standard for, say, a donated kidney or heart is called static cold storage. It's much simpler. You flush the organ with a cold preservation solution to get the blood out, and then you basically pack it on ice, keeping it around 4 degrees Celsius.
Just above freezing, like a fridge.
Pretty much. It doesn't freeze the organ, it just dramatically slows down its metabolic rate, reduces its need for oxygen, slows the buildup of toxic waste products. It puts the organ in metabolic slow motion, but it doesn't stop time.
And the big drawback there is the clock is still ticking. Fast.
Very fast. That's the critical limitation. Its effectiveness is measured in hours, not days or weeks. For a kidney, scattered cold storage might give you 24 hours, maybe a bit more sometimes.
Okay.
But for a heart, which uses a lot more energy, you're looking at maybe just 4 hours.
4 hours. That's incredibly tight.
It's the single biggest bottleneck in organ transplantation today. That short time window severely limits how far an organ can travel. It restricts the time available to find the best possible immune match for the recipient, especially if you want to do more advanced genetic testing, which takes time.
Right.
And it often forces transplant surgeries into being emergency procedures, middle-of-the-night rushes instead of planned operations. That adds complexity, risk, stress for everyone.
Which brings us back to that core challenge underlying both slow freezing and vitrification attempts. The cryoprotective agents, the CPAs, things like DMSO, glycerol, you need them to fight the ice, but they're also toxic, especially at the high levels needed for vitrification or to get into large tissues. That toxicity seems like a fundamental wall researchers keep hitting.
It really is. It's this constant balancing act. Enough CPA to prevent ice, but not so much that it kills the cells through toxicity. Finding that sweet spot, especially for complex organs, is incredibly difficult.
So, given all these challenges, ice, toxicity, the limits of just keeping things cold, where do we actually stand today? The review paper dives into this, looking at different tissues. It seems like a real mixed bag, doesn't it? Success really depends on what you're trying to freeze.
Absolutely. It's heavily dependent on the size, the complexity, the types of cells involved. Simpler, smaller things or tissues that are more uniform. They tend to do much better with current methods than big, complicated organs.
Let's talk about some of the wins first. Where is cryopreservation more established, maybe even used routinely in clinics or biobanks? That review paper lays out quite a few examples in its conclusions and section 2.
Right. One area that's seen real clinical success is ovarian tissue cryopreservation. This is pretty well established now, mainly for preserving fertility in women, often young women, who are about to undergo cancer treatments that could damage their ovaries.
And it works. People have had babies after this.
Yes, that's the concrete proof. There are many documented live births now after cryopreserved ovarian tissue was thawed and grafted back onto the patient. Hormonal function returns, and they can conceive naturally or through IVF. It's a fantastic option.
That's amazing. Are there limitations, though?
Well, the success is mainly with freezing small strips of the outer layer of the ovary, the cortex, which is where most of the eggs are. Trying to freeze a whole ovary is still really difficult because of its size and the risk of damage from lack of blood supply, ischemia, after it's transplanted back.
Okay. What about for males? Testicular tissue.
Immature testicular tissue, ITT, is another area where cryopreservation is offered, mainly in biobanks. This is for young boys facing similar fertility-threatening treatments before they start producing sperm.
So preserving the stem cells.
Exactly, the spermatogonial stem cells. Research shows that preserving small pieces of the tissue itself, maybe around 3 cubic millimeters, keeps those stem cells more viable than trying to freeze them as a cell suspension. Slow freezing is common here.
But is it clinically proven, like ovarian tissue?
Not quite yet. While there have been successful animal studies, like scientists have generated live piglets using this method, it's still considered experimental in humans. There haven't been any reported human live births resulting from thawed ITT yet. So, promising, yes. Available in biobanks, yes. But not fully validated clinically in the same way. The small sample size definitely helps the preservation itself work better, though.
Got it. Moving away from fertility, what about structural parts like heart valves or blood vessels?
Heart valves are a big success story for biobanking. Cryopreserving donated human heart valves is a standard procedure now, and they have very good survival rates and work well clinically when transplanted.
How did they get so good at that?
Standardization helped a lot. Things like the guidelines used by European tissue banks. They did rigorous testing, mechanical tests, structural tests on the valves after cryopreservation and thawing. And based on that data, they've actually been able to significantly extend how long these valves and also cryopreserved blood vessels can be stored, sometimes doubling the expiration date while still being confident they're safe and effective.
That's great. And blood vessels themselves.
Yes, vascular tissue grafts, or allografts from donors, are also commonly cryopreserved. They're used quite widely in surgeries like bypasses or fixing damaged arteries. Many biobanks have standardized protocols.
Do they work as well as fresh?
They generally show pretty good survival rates. But there are still challenges. If the cryopreservation or thawing isn't done perfectly, there can be problems, like the graft rupturing later. DMSO is often the CPA used. But again, adopting standardized protocols, like those in the US and Europe, has really improved clinical outcomes over time. You can even find studies reporting specific success rates, like 5-year survival in the 60s or 70% range for complex aortic surgeries using these grafts.
Okay. What about fat, adipose tissue? People use that for grafting.
Adipose tissue cryopreservation is another well-established procedure in biobanking. It's a really practical option for fat grafting in cosmetic or reconstructive surgery. You harvest fat once, freeze it, and then you can use it for multiple injection sessions later without needing more harvesting.
Convenient. Does it survive well?
Clinical studies show good success in lots of patients, usually with minimal issues if it's properly cryopreserved. The catch is, if you use uncontrolled or just bad freezing protocols, you can run into complications, infections, lumps, bruising, even tissue death. So finding the best way to freeze it is still important. People usually store it at -196°C. DMSO is a common CPA, sometimes mixed with other things like Trailose, which might let you use less DMSO and reduce toxicity. Both controlled slow freezing and simpler, faster, uncontrolled freezing methods are used.
Seems like a theme. Smaller, simpler tissues, or where structure is key, but maybe not every single cell needs to be perfect. That's where we see more success. Any others like that?
Yeah, a few more. The paper mentions the amniotic membrane from the placenta. It's used as a biological bandage or scaffold in wound healing and eye surgery. It can be preserved well cryogenically, although the common method using glycerol at -80°C might cause some cell damage.
In bone.
Bone cryopreservation is established too. Both the patient's own bone, autologous, and donor bone, allogenic. It's particularly used in skull reconstruction, cranioplasty. Like, if someone needs part of their skull removed temporarily, they can freeze that bone flap and put it back later. Allogenic or donor bone doesn't always integrate as well as fresh bone, but it's widely used.
And skin, for burns.
Right. Skin cryopreservation is definitely used, especially in burn centers, for temporary wound coverage. You can cryopreserve skin while keeping the cells viable if you do it really quickly after donation. But often, a simpler, cheaper method is used where the skin is preserved in glycerol at higher temperatures. That skin isn't viable, the cells are dead, but it still works great as a biological dressing or in certain grafting techniques. It's very useful even without living cells.
So looking at all these ovarian tissue, valves, vessels, fat, amnion, bone, skin, the pattern seems clear. Relatively simpler structures, smaller samples, or cases where perfect cell-by-cell viability isn't the absolute must-have, that's where cryopreservation has made real inroads into clinical practice and biobanking.
Exactly. Those are the areas where the existing tools, slow freezing or sometimes vitrification for very small things, can be applied effectively.
Which, naturally, brings us to the flip side, the really tough nuts to crack. The tissues and organs where cryopreservation's success is still very limited, or basically non-existent for clinical use. And it's usually because they're big, complex, and need almost perfect function after thawing. The review paper goes into these too.
This is where we run smack into those fundamental limits of ice damage, CPA toxicity, and just the sheer physics of cooling and warming large, complex objects uniformly. The whole heart is maybe the prime example.
The holy grail, almost.
You could say that, but the review is blunt. The heart has not yet shown signs of successful cryopreservation for transplant. It's incredibly sensitive, high metabolism, multiple tissue types reacting differently to cold and CPAs.
Any successes at all?
Early studies, like decades ago, managed to get small animal hearts beating again after freezing them down to maybe -30°C, but colder than that. Resuscitation was impossible. The risk of cryo injury, especially to the tiny blood vessels and muscle fibers, is just massive. People are looking at vitrification plus things like nanowarming, but applying that to a human heart, the individual variations, the complex structure, it would need incredibly precise, personalized protocols. We're not there yet.
Okay, what about pancreatic islets? Those are smaller, just cell clusters. Why are they still difficult?
You'd think they'd be easier, right? And you can cryogenically bank pancreatic islets, the clusters that make insulin. It's done. But the big problem is low post-thaw recovery. Too many islets get damaged or die during the freeze-thaw process.
So not enough survive to do the job.
Pretty much. There's debate about whether slow freezing or vitrification is better. Using tricks like adding the CPAs step by step seems to help a bit. But overall, the yield and function are just too low right now for cryopreserved islets to reliably make someone insulin independent after transplant. Fresh islets work much better, but they're scarce.
The cornea. That seems thin and relatively simple. Why is that hard?
The cornea is surprisingly tricky, particularly the innermost layer, the endothelium. It's a single layer of cells that's crucial for keeping the cornea clear, and it's extremely fragile and easily damaged by freezing.
And it can't repair itself well.
Exactly.
Yeah.
Corneal endothelial cells have very limited ability to regenerate, so any damage from cryopreservation is usually permanent. That's why eye banks just don't routinely freeze corneas. They use fresh ones stored short-term at 4 degrees C.
Okay, now the really big ones. Kidneys and livers. The organs most people associate with transplantation. Where are we with those?
Still facing enormous challenges. For whole kidneys, success for transplantation is very limited. Standard slow freezing just doesn't work for something that large and complex. It fails. Trying to cool it really fast, like plunging it in liquid nitrogen, it cracks from thermal stress. Yeah. Vitrification seems like the only plausible route. But the engineering hurdles are immense, getting the CPAs evenly into that big, dense structure. And then, the killer. Warming it back up. You need perfectly uniform, ultra-rapid warming through the entire kidney instantly to avoid that lethal devitrification ice.
How do you do that for something fist-sized?
That's the million-dollar question. People have successfully vitrified tiny embryonic kidneys, even transplanted them into mice. But scaling that up to an adult human kidney, the heat transfer, the mass transfer, it's incredibly hard.
And the liver, even bigger and more complex.
Even tougher, generally. Whole liver preservation runs into all the same problems as the kidney, but amplified. It's larger, has a hugely complex blood vessel network, very high metabolic rate. So the challenges of getting CPAs in, avoiding toxicity, preventing ice, and especially achieving uniform rapid rewarming are even more severe.
Any progress there?
There's research showing success, like vitrifying and rewarming a rat liver using nanowarming. But the review notes that scaling that kind of thing to human organs is limited by the sheer amount of materials needed, the complexity, the cost. Some groups are exploring other ideas, like directional freezing for pig livers, trying to control ice growth in a specific pattern. But clinical application, still a long way off.
What about cartilage, or really complex things like fingers or limbs?
Cartilage has various cryopreservation techniques developed, but the review says they aren't widely used in biobanks yet. DMSO is a common CPA. Nanowarming shows some promise for vitrified cartilage, preserving more cells and properties. For complex tissues like digits or whole limbs, things with bone, muscle, nerves, skin, vessels all together, there's much less successful research compared to simpler tissues.
But have digits been cryopreserved successfully?
There are reports in the literature of successful digit cryopreservation, yes. But the review emphasizes it has not become a widespread clinical technique for replantation. It's extremely difficult. Animal studies, like successfully cryopreserving and transplanting a rat hind limb using advanced methods, show it's theoretically possible, but the complexity is huge.
Yeah.
Interestingly, sometimes even just the cryopreserved skin from a fingertip can be useful for reconstruction, even if the whole digit can't be saved.
And other tissues, brain, nerves, muscle.
Brain tissue cryopreservation is becoming more common for research purposes, saving tissue for later study. It shows some promise, but again, the brain's complexity is a massive hurdle. Nerve tissue has shown some successful results in animal transplants, and some studies have shown that muscle tissue and complex groin flaps used in reconstructive surgery can remain viable after cryopreservation. So research is happening across the board, but clinical translation for these complex tissues is slow.
Okay, so if we pull all that together, it seems like we're pretty capable with smaller, simpler tissues, sometimes even getting them into regular clinical use. But for the big, complex, life-sustaining organs or composite tissues like limbs, the old methods just hit a wall because of ice, toxicity, and the sheer scale of the problem.
That really is the crux of it. Based on that scientific review, the traditional approaches, slow freezing and vitrification, despite decades of refinement, face fundamental physical and chemical limitations when you try to apply them to something like a human kidney or heart.
Which means if we ever want to bank organs long-term, truly overcome those hours-long limits of static cold storage, we need something new. New ideas, new technologies.
Exactly. The current toolbox isn't quite up to the job for organs.
But the good news is, researchers aren't just throwing up their hands. There is intense work going on globally to tackle these exact problems. The review paper points to several really promising directions where scientists are pushing the envelope.
They absolutely are. People are attacking this from multiple angles. One area is developing better revival cocktails, special solutions used during thawing that help cells recover from the stress and repair some of the non-lethal damage.
Like a recovery drink for cells.
Sort of, yeah. And there's work on standardizing protocols better, for storage, for transport, to make results more consistent, trying to push those safe storage limits even further. But the big targets remain the same. Stop destructive ice, whether during cooling or warming, and minimize all those cryo-injury mechanisms especially CPA toxicity and osmotic shock.
The paper highlights some really clever potential solutions. What are some of the ones that stand out?
One that gets a lot of buzz is nanowarming. This is aimed right at that critical rewarming problem, especially for vitrified samples.
The problem being you can't warm big things evenly from the outside.
Right. Heat just doesn't travel fast enough through a glassy or icy solid, so the outside warms up too fast, maybe devitrifies, while the inside lags behind. Bad news. Nanowarming tries to fix this by mixing tiny magnetic nanoparticles into the cryoprotective solution before vitrification.
Magnetic nanoparticles
Right.
inside the organ.
Distributed throughout the solution, bathing the cells and tissues. Then, after vitrification, you put the whole sample in an alternating magnetic field.
And the nanoparticles heat up.
Exactly. The magnetic field makes them generate heat rapidly and, crucially, uniformly throughout the entire volume. It's like having millions of tiny heaters embedded everywhere, all turning on at once.
So you get fast, even warming from the inside out.
That's the idea.
Yeah.
Ultra-rapid, uniform rewarming. This is essential to prevent devitrification in vitrified samples, and it might even allow using slightly lower, less toxic CPA concentrations in the first place because you know you can rewarm it fast enough.
Has it worked?
It's shown real promise in animal studies, successfully rewarming vitrified rat kidneys and livers, preserving their structure and function. Also worked with pig arteries and heart valves. But there are potential downsides. Some kidney studies suggest that the rapid heating might actually damage the very delicate capillary network. And scaling it up safely and reliably to human organs is still a big challenge. Getting the nanoparticles distributed perfectly, making sure they're safe and don't cause other problems, plus the cost for large volumes, lots of hurdles still.
Okay, so nanowarming tackles rewarming. Are there other approaches, maybe inspired by nature, or ways to work with ice instead of just fighting it?
Yes, definitely. The idea of controlled partial ice freezing is interesting. It's bio-inspired. Think of wood frogs. They can actually survive freezing partially solid with ice in their extracellular spaces.
How do they do that?
They have natural cryoprotectants, but this research explores using special biocompatible ice nucleators. These are substances that encourage ice to form in specific ways at specific locations during slow cooling at relatively high subzero temperatures, like not down at -196°C. The goal is to guide the ice formation into patterns that are less damaging to the cells.
So manage the ice, make it form where it does less harm.
Kind of like that. It's shown some early success in models like blood vessels, and people are looking at it for complex tissues like lungs. It's a different philosophy. Not zero ice, but maybe tolerable, controlled ice.
And what about tackling that core CPA toxicity problem directly?
Several strategies there. One is called liquidus tracking. It's pretty sophisticated. The idea is to add the CPAs gradually, in steps, while you're cooling the sample. You carefully track the liquidus curve, that's the temperature at which freezing would start for the CPA concentration you currently have.
So you keep adding CPA just enough to stay ahead of freezing as it gets colder?
Exactly. You stay just above the equilibrium freezing point for the current concentration. This might let you prevent ice without needing that super fast cooling of vitrification and potentially use lower peak CPA concentrations overall, thus reducing toxicity. Then you reverse the process during warming, removing CPAs gradually to minimize osmotic shock. It needs very precise control, but it's promising.
Clever. Any other ways to reduce toxicity?
People are looking at using non-Newtonian fluids whose viscosity changes with shear force, or rheomagnetic fluids whose viscosity changes in a magnetic field. The idea is maybe these could help load and unload CPAs faster, reducing exposure time. Or perhaps create a kind of pseudo-vitrified state at less extreme cold, maybe -60 or 70°C, by making the solution super viscous, which could inhibit ice with lower CPA levels. Still quite experimental.
And then there was that really different one, isochoric cryopreservation, using pressure.
Yeah, isochoric is fascinating. Isochoric just means constant volume. You put the sample in a strong, rigid, completely sealed container, then you cool it below zero.
But doesn't water expand when it freezes inside a sealed box?
It does, and that's the key. As some water starts to freeze and expand inside that fixed volume, it builds up enormous pressure.
Yeah.
And high pressure changes the freezing point of water. It lowers it,
So the pressure itself stops the rest of the water from freezing.
or at least suppresses ice formation in the bulk liquid where the sample is. Ice might form in specific areas, but the high pressure keeps the sample itself liquid and ice-free,
Yeah.
even at temperatures significantly below 0°C, at normal atmospheric pressure, maybe down to -20°C or even lower.
Wow. And the potential benefit?
Huge potential benefits. You might need minimal or even no CPAs at all. completely avoiding the toxicity issue. And because the pressure effect works throughout the volume, it could be suitable for larger tissues and organs. It might also make vitrification easier by suppressing random ice formation, maybe allowing lower CPA concentrations if you combine it with vitrification methods. It's a really neat thermodynamic trick. Still needs development, but very intriguing.
Okay, those are some really creative, futuristic ideas bubbling up in the labs. Nanowarming, controlled ice, pressure tricks. But this is where the story takes a sharp turn towards the present with that news article. Because while those are mostly still research concepts, one company, X-Therma, seems to have actually built something that works now for preserving a whole organ. And their mission, it's incredibly ambitious to abolish the organ transplant list.
Yeah, it's a bold claim, but the approach they took is really interesting. They didn't just try to slightly improve the old CPAs like DMSO with all their known toxicity issues. They went back to nature.
Like the wood frog.
Sort of, but more like fish and insects that live in freezing water. They study the natural antifreeze proteins these creatures use to survive sub-zero temperatures without freezing solid. But instead of just using those proteins, they used biomimetic chemistry, basically, mimicking biology to design and build a completely new molecule. It's not a protein, it's called a peptoid.
A peptoid, what's special about it?
The article says this engineered peptoid is designed to be better than the natural antifreeze proteins. Maybe more stable, easier to make, less likely to cause an immune reaction, but crucially, having superior ice-controlling properties.
Okay, so what does this peptoid do to ice that's different?
Its main trick, according to X-Therma and the reports, is preventing recrystallization. Remember how small ice crystals can grow into big, damaging ones?
Yeah, during warming or if the temperature fluctuates.
This peptoid is designed to stick to the surface of any tiny ice crystals that might start to form. It basically latches onto the crystal faces and blocks more water molecules from joining, stopping the crystal from growing. So, as their CEO puts it, their ice does not damage cells because the peptoid keeps any ice that forms incredibly small and stable.
So it doesn't necessarily stop all ice from forming, but it controls it, keeps it harmless even below zero.
That seems to be the key mechanism. It allows preservation below 0 degrees Celsius, but without letting those destructive large ice crystals run rampant.
And they've built this into a product.
Yes. This peptoid is the star ingredient in their preservation solution called X-Vivo. It's specifically designed for organs. The article points out some key properties. It's apparently non-toxic at the concentrations they need for organs, which is a huge difference from the high levels needed for traditional vitrification. It's also serum-free and protein-free, which helps with purity and safety.
And its main function.
Its unique function, thanks to the peptoid, is enabling ice-free preservation specifically in that crucial temperature range between 0 degrees C and about minus 20 degrees C.
Sub-zero, but not deep freeze. Why is that range important?
It's potentially a sweet spot. It's cold enough to slow metabolism way down, much more than the 4 degrees C of static cold storage, buying lots more time. But it avoids the extreme cold of liquid nitrogen, which might sidestep some of the deep cold toxicity issues or the physical stresses like fracturing that can happen with vitrification at minus 196 C. The X-Vivo solution is designed to be flushed through the organ, replacing blood, keeping metabolism low, and protecting it during transport.
And they have a special container for this, too.
Right. They developed the time seal device. It's basically a smart cooler, portable, specifically designed for static preservation of adult donor kidneys using the X-Vivo solution. It has sensors built in to monitor temperature, how long it's been stored, even its location, using GPS, presumably. But the really big deal for logistics is that it's designed to be carried on commercial flights.
Wow. Not special medical transport.
Apparently not always necessary. And crucially, the article stresses this. Unlike some complex machine perfusion systems that actively pump fluid through the organ, the time seal device needs no external power, no blood supply, no oxygen tanks during transport. You flush the organ with X-Vivo, put it in the time seal it, and you're good to go.
That simplifies things massively.
Hugely. Just think about the logistics of getting power and specialized equipment onto any plane or ambulance versus just carrying a self-contained box.
Incredible. And this isn't just theoretical. They've got regulatory recognition.
Yes, that's a really significant milestone. Back in May 2022, the FDA granted X-Therma's X-Vivo solution and the time seal device its breakthrough device designation.
What does that mean exactly?
It's a big deal.
Yeah.
The FDA gives this status to technologies that can provide more effective treatment for life-threatening conditions and represent a significant potential improvement over existing options. For X-Therma, The designation was specifically for preserving human kidneys for up to 120 hours.
120 hours, that's 5 days.
Days. Compare that to the current 24 hours or less for static cold storage. It signals that the FDA sees serious clinical potential here. This isn't just a lab curiosity.
And then came the big demonstration described in that October 2024 news article, the transatlantic flight.
Right, this was the real showstopper. A team led by Dr. Gerald Brandecker, he's a top transplant surgeon, and researcher at Johns Hopkins did this groundbreaking experiment. They took a pig kidney.
Why pig?
Pig organs are very similar physiologically to human organs, so they're often used as the final step in preclinical research before human trials. They preserve this pig kidney using ex vivo in the time-sealed device, keeping it at a controlled sub-zero temperature. Then they put it on a regular commercial passenger flight from Baltimore across the Atlantic to Innsbruck, Austria.
Just on a plane. In the cargo hold, presumably?
Presumably, yeah. The key is it didn't need special handling beyond what a commercial flight could offer. And the kidney was preserved like this for about 48 to 72 hours during the whole process.
2 to 3 days, sub-zero, flying across the ocean. What happened when it got there? Did it survive?
The results reported were extremely positive. When it arrived in Innsbruck, the team there checked its viability. They hooked it up to a machine perfusion device standard practice to assess organ health, and the kidney showed it was viable and functional.
Just one kidney, or did they do more?
They weren't just lucky once. They repeated this whole transatlantic trip and assessment 5 times with 5 different pig kidneys, all preserved for similar 48, 72-hour periods. And they got consistent positive results each time.
Okay, consistency is key. But the ultimate test is transplanting it, right?
Exactly. And that was the real proof of concept, the headline result. They took one of these kidneys after it had been preserved ice-free, sub-zero, for a full 72 hours using ex vivo and time seal, transported transatlantic, and they transplanted it into a recipient pig. The article states that the transplanted kidney provided life-supporting function and maintained normal kidney function in that pig for 200 days. That was the entire length of the study's observation period.
Normal function for almost 7 months, after being effectively on ice, sub-zero for 3 days and flown thousands of miles.
That's what the report says. It's genuinely remarkable. It's a massive leap beyond that 24-hour limit for kidneys with current static cold storage.
The implications of that seem huge.
Transformative is probably the right word. This directly tackles that biggest bottleneck, time. Current methods basically limit transplants to a regional affair, or require incredibly complex, fast logistics. If you can reliably extend that window to 48 hours, 72 hours, maybe even that 120 hours the FDA mentioned, it changes everything.
How so?
Organs aren't geographically tethered anymore. A donor in one state, or even one country, could potentially provide an organ for someone much, much farther away. This could directly help close that huge gap between organ supply and patient demand. More organs get used.
And fewer get discarded. You mentioned that was a problem.
A big problem. Estimates for over 20% of kidneys recovered from deceased donors in the U.S. were discarded in 2021. Often just because time ran out to find a match or get the organ there. More time means fewer discards. It also means more time for better matching. Maybe doing more advanced genetic tests to find the absolute best immunological match, which could reduce rejection rates and improve long-term outcomes.
Makes sense. Better matches take time.
Right. It expands the potential donor pool geographically. And it could make transplantation less of an emergency scramble, allowing for more planned, scheduled surgeries, which is generally safer and less stressful for everyone involved. It just makes organs more available globally.
And Dr. Branneker, the surgeon involved, seems convinced.
Very much so. He's quoted calling the X-Therma tech streamlined, modifiable, and easy to use. He loves the practicality, no extra steps, no need for complex perfusion machines during transport. That self-contained, no power needed aspect is huge for real-world use. He believes it could fundamentally change the ways of organ preservation. Coming from someone in his position, that carries a lot of weight.
And this isn't just company PR, right? They're sharing this with other scientists.
Absolutely.
Yeah.
The article mentions these results from the transatlantic trips and the underlying studies have been presented at major scientific meetings. The Society for Cryobiology, the Transplantation Society. That means peer review, scrutiny from other experts in the field. It adds significant credibility. So let's wrap this up. What have we seen on this deep dive? We started with the basic science, right? The huge challenges of ice damage, CPA toxicity, problems that have really held back organ preservation for decades.
Right. We saw why traditional methods like slow freezing and vitrification work okay for some simpler tissues, but hit a wall with complex organs.
Then we looked at some of the really cutting-edge research trying to break through those walls, nanowarming, to solve the rewarming puzzle. Clever ways to manage ice or reduce CPA toxicity like liquid extracting, even using pressure with isochoric methods. Really smart science happening there.
Futuristic stuff.
Futuristic, but maybe not too far off for some things. And then we landed on this very tangible breakthrough from X-Therma, demonstrating they can keep a complex organ, a kidney, viable at sub-zero temperatures for days, fly it across an ocean on a commercial plane, and have it work after transplantation. That feels like a shift from lab concepts towards something that could actually reach clinics relatively soon.
And that's the really exciting part, isn't it? This isn't just abstract science anymore. It's moving towards practical solutions that could have a massive impact. Being able to preserve organs longer, transport them farther. It's not just a technical feat. It genuinely offers hope for saving more lives, improving transplant outcomes, making the whole system work better for patients waiting desperately.
Yeah, the potential impact is global. It could help make transplantation more accessible, more equitable. And while this big breakthrough was with kidneys, you have to wonder, could this peptoid technology, this approach of controlling ice in that sub-zero range, eventually work for other organs, other complex tissues? It opens up so many possibilities for future research and therapies.
So here's something to think about as we finish. Just imagine a future, and maybe it's not that far away now, where the distance between a generous organ donor and the person who needs that organ just doesn't matter anymore, or matters much, much less. What does that future look like? What possibilities does it unlock, not just for kidneys, but for hearts, livers, lungs, maybe even those complex limb transplants? What does it mean for health worldwide? We've really only dipped our toes into the deep, cold waters of cryopreservation today, but hopefully you got a better sense of the incredible challenges, the ingenious science being done, and the truly exciting breakthroughs that are starting to emerge in this vital field.